All right. Thank you, everyone, for making time today. As Sarah indicated, I'll walk through some proposed strategies around compositional identity profiling and the corresponding risk management activities that many of you perform on a daily basis. So one of the challenges, I think, when it comes to characterizing substances is to obtain mass balance type qualification of materials. It helps to ensure process reproducibility, of course, contaminant control, as well as if your products within certain regulated industries, some contaminants or impurities, there are regulatory limits imposed upon those particular substances. And so as a consequence, the effort to seek what I would call a composite identity, right? An identification of all components observed in a substance, in a material that's produced in modern manufacturing processes. Obtaining that composite identity, the identity of individual components observed, allows one to perform chemical structure-based risk assessment comprehensively. Of course, there's also structure to material property assessment considerations. So if, for example, you have impurities that impart a particular effect in the substance's quality or performance over time, understanding what that structure to material property relationship is and make the corresponding assessment. And then, of course, structure-based process controls. If you can understand the source of a particular component based on its identity, you have the ability to determine where in a process that material is being produced and establish a control around it. What I think is important is also the decision support needs that you need to satisfy here. Efficient identity determination processes, the process by which you identify a component in a substance has to be efficient. We'll talk a little bit about that later. Also gives you the ability to perform effective risk management. If you understand composition, the source of individual components, how you're controlling them, also based on the identity, what potential risk the presence of a particular material or substance may impart, it allows folks to have effective risk management. And then finally, in building these things and having an effective decision support interface, you can perform like chemical-based industrial forensics. And we'll talk a little bit about those types of forensics activities a little later. So to illustrate the process of identifying a so-called composite identity on a substance, let's consider a relatively simple process, aspirin synthesis. And from this relatively simple process, we have a set of materials that are transformed through operations, right? A unit operation. So over even a simple cycle, you can establish quality attributes based on the materials, the composition of those materials, how they are transformed through a set of operational steps. Establishing mass balance at each stage of the process can be essential for understanding the final product's overall critical quality attributes. An illustration of how that would look is a so-called process map. And what you can see here is a synthetic route whose composition is like nominally displayed on the screen, right? So in the first stage, you have your starting material, which is phenol, but it may be important to note that a particular impurity may be associated with that starting material. And then as you go through your unit operations, you can see potentially, in this slide, for example, at stage 2, you can see the dimethylphenol being transformed as a function of the transformative unit operation to another synthetic component. So what you have here is the ability to map out the entire process is like composite composition, okay? And what that ultimately gives you is identities for each of the potential substances you might observe it in the final material. So just some additional notes here. when it comes to that component identity lifecycle, characterizing a substance through chromatographic or spectroscopic means, identifying components observed through that process based on spectral characterization, and understanding the scalability that at industrial scale I think is important. So this this is a nice depiction of the identity life cycle. You get chromatograms through HPLC with UV detection. Upon detection and integration of individual peaks in those chromatograms, you may also obtain mass spec data to get initial confirmation of, let's say, molecular weight and molecular formula, and based on fragmentation pattern, some sort of partial structure information. Over time, as you collect more spectroscopic information, that may reveal structures, and then I'm sure confirmation of those structures through independent synthesis, reference material acquisition, and spectral comparison are all part of that cycle. Thinking about that process in some detail, you start in the original chromatograms with peaks, right? And the only attribute you have in the first run of or characterization step is relative retention time, and you build that up over time. So as an example, collecting spectral information and associating that spectral information, even when it's still nominal identity information like relative retention time, being able to build that up in a life cycle type fashion is often important. What you may end up getting then is being able to take one of those components and associate spectroscopic data that you acquire over time. And an illustration of that, something like this, where you have independently acquire data, but making that association within your digital application is often important. And just some examples from those of you that know ACDLabs, our ability to make those associations within our Spectris platform is a capability you have. From there, you can associate candidate structures, or would want to associate candidate structures in this digital interface. And so in this case, peak number 6, being able to associate a Marcouche representation of that structure. You may not know its discrete structure, but you know it may have these specific structural attributes. Having that ability is very important. And then from there, once you have final structure, being able to go back and confirm in additional lots of materials produced, being able to confirm through spectral comparative analysis. Here's an MS assignment based on isotope pattern and fragment pattern, revealing an excellent match between the proposed structure and the corresponding spectroscopic data. Just to show the blow up here. But if we look at that life cycle, the life cycle figure that we talked about before, you have to repeat that for all of your components observed in the materials you produce, and then repeat that across all of your components for all of your manufacturing processes. So I guess to illustrate the point that whatever digital software applications, data management applications you bring in, they have to perform at scale. And that's an important consideration when implementing new technologies to support such efforts. An additional consideration is organizing data with the experimental context and the data in the same system, right? So within our Luminata application, what some of you may be familiar with is the ability to build these so-called project map. In addition to the process mapping we showed earlier, the ability to relate that process map to all of the other, call them characterizing or manufacturing processes that you might have. Being able to digitally characterize your both experimental context and your manufacturing context is important. So just to show an illustration of that digital contextualization capability, here's a degradation study represented by a set of exposure conditions, collection time points, etc. Being able to then map an individual stage like humidity with 3 different pull points is important. But what that then yields is an overarching context for all of your different characterization experiments. So this is a project map with different levels in the various experiments, and then some sort of indication of readiness or completeness. So that's what the different symbols in the blocks represent. I'm done with photo, but my thermal stability assessment is still underway, and I haven't started vaporization or humidity yet, just as an example. So back to that impact and risk assessment. If a material is produced, and this might be a familiar looking chemical structure for some of you, you may want to ask the question, where else have I seen this molecule? This gets to extensibility. And if you build such digital systems, it gives you the ability to look and find, where else have I seen this molecule? So one attribute of our Luminata platform is the ability to perform that cross-experiment risk assessment. So if it's determined that this particular substance needs to be controlled, and there needs to be an assessment of where else in my processes might this compound be observed, this is a feature where I can pivot that digital system and look for a specific compound across processes, right? So this is a reaction map that we referred to earlier, but then I can also look in my batch records as well to determine if that material is possible to be present and have action plans that correspond to that potential presence. Furthermore, comprehensive risk assessment, as a summary, being able to perform that composite identity lifecycle in an efficient fashion, being able to understand composition when it is mediated by your processes, right, and having a depiction of that process mediated identity in a process map is often important. And then being able to perform read across, right, similar to the pivotability, looking for a single compound. You want also the ability is for a given substance. In this case, if I have a process mediated identity, composite identity, what are the properties of those individual compounds, and being able to display that in one view and really get a comprehensive read across of risk assessment in one interface, and pivotable interface, I think, is the important point here. And that's it in summary. I'm happy to answer questions or discuss additional topics in detail as you like. Thanks, Sarah. I guess we can open the floor up to questions.
Absolutely. All right. So a reminder, everyone, you can ask your questions in the panel on the right. So we have a few to start. So risk assessment involves bringing together an incredible amount of data, which is then used by many people. So how do you and ACDLabs think about designing digital tools that cover such a broad area?
Wonderful. It's a great question. So I would argue three critical considerations. One is that oftentimes the variety of data is the format of the different variety of data that comes in. And the data can be certainly instrumentally derived or experimentally derived. And so you need a system that handles the breadth and depth of those different types of data, number one. Number two is, let's call it human versus robot association of data to the right places. So at ACDLabs, as you may all know, we have software interfaces that allow for import, view, process, analysis of different types of analytical data, all in a single interface, and that's our Spectrus processor product. You may also be aware of one of our newer products called Spectrus JS, that is a browser-based processing application, starting with NMR, but working on other techniques as part of our development roadmap. Allowing users to conveniently and open up data that they have access to and interact with it themselves. We also have an automation platform that allows, with the appropriate implementation strategy and processing strategy, being able to marshal data from the source to convenient locations. And one of the trends we see in various industries is moving data from the source to the cloud, where presumably everyone can access it. If you're interested in this subject of so-called effective analytical data management, I'd encourage you all to check out a webinar provided by our our customers over at Pfizer, they've spent a lot of time thinking about how to implement the most effective strategy for analytical data management. For a long time, even when I was there 20 years ago. So happy to discuss in more detail with whoever asked the question.
Okay. We have another one here. Can Luminata expect the degradants production depending on different temperatures and different humidity conditions?
So expect, I guess, is the part I'm not sure about. When it comes to handling data, if you're sampling at different temperatures and different exposure conditions over time, right? If you're performing some sort of kinetic stability analysis, yes, the system for the process mapping system is configurable and user configurable. So you can define for yourself what time points and where in a stability assessment you want to associate data to. So that process map is configurable, number one. For folks that are intending to predict the likelihood of a particular degradant appearing, we do integrate with the LASA platform. We've done that for a variety of customers, so you can bring in your predicted degradants. I'm not sure, frankly speaking, how and whether there is tunability of those prediction modules from LASA. Predict a set of structures for a specific time point. I know they have condition-specific prediction, but I'm not sure about time, to be frank.
Okay. All right. And then a bit of a broader one here. So your talk was mainly focused on pharmaceutical applications,
Mhm.
but you also have experience with consumer packaged goods, and food and beverage firms. So are there concerns about product impurities and contaminants similar, in those institutions? And do you think these sectors can learn from each
Yeah.
other with respect to impurity control?
Certainly, the answer there is yes. For those of you in those industries, you'll recall with interest the degree of control over acrylamide. Right? Industries got together, worked on the so-called acrylamide playbooks. Um, Being able to detect, quantify the amount of acrylamide present in a product, a food product, is really important. Um, Being able to then assure that your products, when they hit the store shelves, do not have acrylamide levels above a certain threshold. I worked at PepsiCo. I Frito-Lay is a part of PepsiCo. For those of you in the UK, Walker's Crisp is a part of PepsiCo as well. And so as part of your manufacturing process for even things as supposedly straightforward as potato crisps or chips here in the States, um, being able to assure that acrylamide levels don't exceed a certain threshold is a really important consideration. And so all of those methods of analysis, um, interwoven with the digital context. It's not a synthetic process in this case, it's a cooking process. Right? And so characterizing how acrylamide evolves over different frying conditions, the quality of the oil that you use, characterizing that quality to ensure the appropriate energetics are applied during the cooking process. All of those are important. And so similarly, being able to represent those processes in a digital construct, but also associate the characterization data, ultimately gives you effective decision support and risk mitigation, risk management, um, in a CPG or food and beverage context, just like in pharma. And there are certainly like industry specific considerations. There's modalities that you have to represent that might be different in food and beverage or CPG than in, let's say, small molecule pharma. But I think the digital strategies, the data association strategy, and the contextualization strategy are across industry from my perspective.
Okay. All right. I think we have time for one more really quick question.
Awesome.
Someone asked, does this software, I assume that's Luminata, help in
Mhm.
predicting the correct structure of an impurity when MS and carbon and proton NMR data input is given?
Yeah, so great question. Luminata is a part of our Spectrus platform. And one of the capabilities of that platform indeed is to facilitate effective determination of identity. We would call it structure elucidation or structure confirmation. And indeed, there are a variety of predictive tools and elucidation tools within our platform. Happy, I'm sure, if for whoever answered the or asked the question, happy to follow up with those specific capabilities within the Spectrus platform. But the short answer is yes.